The overall goal of this experiment is to differentiate human-induced pluripotent stem cells into hepatocytes-like cells in a manner that is suitable for high-throughput screening.
Liver cells are difficult to grow in the laboratory. However, they can be generated from induced pluripotent stem cells, which provides a screening platform to identify treatments for liver disease. The main advantage of this technique is that unlimited numbers of hepatocytes can be generated from any patient with liver disease that recapitulate the disease in the laboratory.
The procedure is relatively easy to establish, even without extensive knowledge of stem cell biology. Its reproducibility allows researchers to screen thousands of compounds without the need for complex robotics.
Ray Liu, a postdoctoral fellow, and Paige Lamprecht, a graduate student, will be demonstrating the procedure today.
First, dilute the fusion protein ECAD to 15 micrograms per milliliter with Delbeco's phosphate buffered saline, or DPBS, that contains calcium and magnesium. Coat 100 mm suspension tissue culture dishes with 5 milliliters of diluted matrix and incubate at 37 degrees Celsius for at least 1 hour. Following incubation, remove the substrate and replace with 5 milliliters of iPSC culture medium. Retrieve a vial of cryopreserved human-induced pluripotent stem cells from liquid nitrogen. Thaw at 37 degrees Celsius until a small ice crystal remains. Then, gently pipette the cells into a sterile 15 milliliter conical.